My name is Emily Finn. I'm a postdoc with Peter Bandettini in the section on functional imaging methods. And I'll be talking today about how can we extract individual differences with fMRI, and can we then go on to create biomarkers from the imaging data. So just to start with defining individual differences, usually when we talk about individual differences, we mean ways that we're consistent in our own behavior across time and contexts, while also being reliably different from others. And so we all know from living in society that these things are real, right? They're present in cognition and behavior. We all have our own strengths and weaknesses and idiosyncrasies. And so if you believe that processes of the mind are instantiated in the brain, then you should also believe that these differences are present somewhere in the brain, whether anatomy or function or both. And so the question is not so much whether these are present, but whether we can detect them using the non-invasive imaging tools that we have available to us. So with that definition, I'll give you an outline of my talk today. So the talk will be 3 sections, although the last will be by far the longest, so bear with me. I'll first start by convincing you why we should care about individual differences. Then I'll tell you more or less what we know about individual differences so far. And finally, I'll spend a lot of time talking about where the open questions and controversies are in this line of research. So starting with the first one, why should we care about these individual differences in the brain? I think there's 2 main reasons that we ought to care about this. The first is that it can lead us toward a deeper understanding of cognition. So we're at a point now with human functional imaging where we have a pretty good handle on the sort of general blueprint for human brain function and sort of general maps for different cognitive domains. So for example, if you do oh shoot, the bottom of my slides are cut off, that's unfortunate. There's a citation to neurosynth on the bottom there, but essentially if you do these large-scale meta-analyses of hundreds and thousands of studies and you query things like working memory or language or social cognition, you come up with sort of these general maps that we all in cognitive neuroscience might have been able to draw sort of on our own. But we know that the individual subjects that went into these maps might all look slightly different. So if we blow up this working memory one, we might see that each individual brain actually looks a little bit different while performing a working memory task. And so this tells us that there's a degeneracy here. There's multiple ways to sort of achieve or perform the same type of cognitive function depending on different individuals. And this variability could relate to things like task performance. And within that, we might see some sort of linear, at least monotonic relationships, such that more activity is associated with better task performance. There may even be nonlinear effects or expert effects. This variability could be associated with different cognitive strategies or styles. So even given the same task performance, are people using different strategies to arrive at that performance level? And finally, we know that there can also be within subject changes, like with learning or practice effects. And so all of these potential explanations or relationships to this variability could be reflected in the brain in a number of ways. We could see univariate or multivariate relationships with activity in different regions. We could see effects in activity or connectivity or other features of the brain. But the point is that I think by getting a stronger handle on mapping from individual brain activity to individual cognitive performance and behaviors, that this will deepen our understanding of cognition itself. The second major reason to care about individual differences is that it can give us insight into mental illness. So if you think about sort of the traditional diagnostic model for a given trait that exists on sort of a bell curve, let's say, in the population, the categorical model of illness will say anyone that's scoring super high or super low on this trait, let's say this is trait anxiety, which we know varies even among the healthy and subclinical population. Maybe people that score at the extreme end here are given a diagnosis of clinical anxiety. So they're called patients, and everybody sort of falling below that cutoff is called a control. And then so classically in a brain imaging experiment, we would stratify our subjects into 2 groups, measure something about the brain, and maybe we observe a mean difference in this hypothetical brain variable between our control and our patient groups. But you can also see that there's a lot of heterogeneity and a lot of overlap between the groups. So what I and many others think is that by moving away from this categorical model toward a more dimensional model of illness where these traits are treated as the spectra that they truly are, and so we preserve this sort of continuous trait here, and we relate this in a parametric way to something about the brain. Even in a study where we don't have access to patients, we might learn more by, even if we just parameterize, so to speak, this part of the curve away from that super high extreme, it might teach us something about what's going wrong once we get to these super extreme values. And the hope here is that by approaching the problem in this way, we can get more power and we can potentially start to build some translational tools based on functional imaging. Okay, so that's sort of a broad overview of why we should care about these differences. So what do we know so far about individual differences in brain function? I'm going to briefly outline this technique of functional connectivity. I think you've probably already heard about it a number of times in this course. And there's many ways and flavors to do functional connectivity, but for at least the first few sections of this talk, I'll be relying a lot on this technique and data analysis approaches that use this, so I'd just like to get us all on the same page. So typically, when I do functional connectivity analysis, I take the brain and carve it up into a set of nodes or regions that cover the cortex, the subcortex, and the cerebellum. And then for a given pair of these nodes, so say this red node here and this blue node, we can look at how strongly correlated their signal fluctuations are over the course of a scan. If we correlate those two, in this case, we see they're fairly tightly coupled in their functional activation. If we do that for all possible pairs of nodes in the brain, we can construct this symmetric, undirected functional connectivity matrix, perhaps better called a functional correlation matrix, since there's no directed information here. We're not necessarily implying a structural connection between these two regions, but we're just observing that their activity tends to fluctuate at more or less a similar pattern throughout the course of our scan. And so we're able to calculate these functional connectivity matrices for individual subjects in individual sessions. And I'm not going to dwell too much on this, but just to sort of go through the study that first got me interested and my colleagues interested in these individual differences, we were curious to see how stable these functional connectivity matrices were in a given subject over time and between different types of cognitive states. And so I'll tell you quickly about this study that sort of started my interest in this field. So this was a few years ago now. We were using data from the Human Connectome project. This was an early release of this data. So we had 126 healthy adults, ages 22 to 35 years old. Interestingly, this data set contained 50 sets of twins. So 100 out of the 126 people actually had a twin in this same data set, which is interesting because it serves to make our question harder. So if there's anyone in the world that we would expect you to look most like, it would probably be your twin. And so our question here was, just to tell you a bit about the HCP scan protocol. So people came in for 2 days of scanning. They had a resting state run on the first day and several tasks, including a working memory task and a motor task. Then they came back for a second day of scanning where they did a second resting state run and some other cognitive tasks, including a language task and an emotion task. So essentially what we did in this first experiment was we calculated these functional connectivity matrices for every subject in each one of these sessions or these task states. And then we were asking the pretty simple question of, can we match up an individual? Can we find their corresponding functional connectivity matrix between two different sessions? So for example, we might try and match connectivity during the resting state between day 1 and day 2, or we might try and match the resting state first day scan to the language scan, and so on and so forth. And when we did this, we observed that we were able to achieve a really high degree of accuracy. So we were able to identify people between 54 and 94% of the time based on the specific pairing of scans that we were using. So some of them did better than others, but overall this was a really striking result because chance here is less than 1%. So if we were just randomly guessing, we would get it right 1 out of 126 times. And here we're seeing that there is really quite a strong individual signal that's embedded somehow into these connectivity matrices, and it's somewhat independent of task state, although I'll come back to that. And when we looked to see which areas of the brain were contributing most to this individual signal, we observed that it was these networks here, so this sort of purple and teal network that have nodes that are concentrated in the higher order association cortices of the brain, so frontal, parietal, and temporal regions. And this was in line with what other people have observed. These are typically the loci of the most inter-individual variation in both structure and function. And so our results align with that, and these networks were the most discriminative of individuals. Now this was an interesting result, so it's telling us sort of you always look most like yourself, regardless of what you're doing while you're being scanned. But this result in and of itself, we don't need to put someone into an MRI scanner to know who they are. We can tell that just by looking at them, right? So the real interesting piece of this will be if there are features of these connectivity profiles that relate to behavior or some sort of real-world output of the brain. And so in this first study, just sort of as a proof of concept of that, we looked to see if we could use these functional connectomes to predict people's levels of fluid intelligence. So fluid intelligence is a construct from psychology, basically people's innate or intrinsic reasoning ability. And so everyone in this data set performed this progressive matrices task outside the scanner, which purports to measure fluid intelligence. And this is something that varies even among the healthy adult population, which is what we had here. So this was the distribution of scores on this test in these 126 people. And we developed a very simple machine learning algorithm, you could call it, that we call connectome-based predictive modeling, or CPM. And the idea of this approach is that we're taking in information from all but one of the subjects. We're building a model relating the strength of the connections in certain parts of this matrix to people's score on this fluid intelligence task. We're building this model on n-1 subjects, and we're testing it on the left-out subject. And we can see how well our model does by correlating the scores that our model predicts versus the scores that our model, that we observe the subjects actually to achieve on this test. And so when we do this using the whole brain, and this is from resting state connectivity, so people are lying in the scanner doing nothing at all, and we're able to predict with some degree of accuracy how well they do on this very challenging test of sophisticated cognition. So this was encouraging in the sense that, you know, these functional connectivity profiles do seem to contain information that's behaviorally meaningful. This graph here is based on a whole brain, basically feeding the model the whole brain connectivity matrix, but when we look to see, again, which of the networks were most contributing to this model, we again saw that it was these sort of frontal parietal temporal regions that were contributing the most predictive edges or connections. Further confirming that the areas that are most discriminative of people are also the most predictive of individual differences in behavior. So since then, we've applied this approach to a number of different other data sets and behaviors. We've gotten some traction on this. So, for example, we've seen that resting state connectivity can be used to predict both levels of sustained attention in healthy adults, but also symptoms of ADHD in children. Work from Peter Bantini's lab done by Dave Jangra here at NIH has extended this to predict people's reading ability from both task-based and resting state scans. We've seen that this approach can predict symptoms of autism as well as even things like personality traits. So all of these predictions are far from perfect, as you can see, so we have a long way to go, I think, before these approaches are ready to be rolled out in the real world. But the crucial thing here is that we're using cross-validation, or in other words, we're building models on a subset of subjects, and we're testing them on brand new subjects to see how well they generalize. And so this, I think, offers some preliminary hope that we might someday be able to derive useful biomarkers based on this type of data. And just to sort of touch back on this point one more time, so trying to localize these individual differences, we weren't certainly the first to start looking at these, so there's already been a lot of evidence that the biggest differences between subjects are found in sort of these most evolutionarily recent regions. So you can see the yellower areas here correspond on the top row to both where the cortex has expanded most in terms of its surface area during evolution, and on the bottom row it's where we see the most intersubject variability. So there's some correspondence there, and certainly you know, it's likely that anatomical differences are playing a large role here. So function is always sort of superimposed on anatomy, and it's difficult to completely tease them apart. And so here, looking at both sulcal depth variability and cortical thickness variability, you can see that there's some overlap with the most intersubject variability in function, although it's not perfect. So teasing apart function and anatomy is challenging, and I think that's an area that people are actively working on, but we'll need some more research before we can do that perfectly. There's also been some really nice work from a group at WashU. So this was a paper where they scanned 10 individuals many, many times, and they had them doing different tasks. So this is somewhat similar in spirit to the work on the Human Connectome Project data, but here it's sort of densely sampling from 10 individuals, and they were able to actually partition the variance in connectivity into factors associated with individuals, tasks, and sessions, and then sort of the interactions. And the point of this slide here is to show that, so the redder the area, the more variance it accounts for. And so you can see this individual map here. Most of the variance in conductivity is accounted for by who the person is. There's a little bit in the interaction here, so sort of who you are and what you're doing. But there's very little that's sort of accounted for completely by task, and there's very sort of minimal session effects. So that again offers some hope that there's some stability here at the individual level. Okay, so we know these individual differences are there, but there's many open questions about sort of what they really mean and how we can best measure them and what we can do with them eventually in a real-world setting. So for the rest of the talk, I'd like to focus on where I see sort of the biggest open questions and controversies. And I should have said this at the beginning, but please feel free to interrupt me with any questions as we go along. Okay, so I think this sort of open space can be roughly divided into three different aspects of our studies. So the first are open questions with data acquisition. Then we have data analysis, and finally we have potential applications. So I'll sort of be going through and building out the questions that I see as most pressing in each of these spaces. And so starting with data acquisition, the first thing we need to do when we design a study is ask who do we actually want to study, right? What should our populations be? And right now we're at an interesting moment in the field because we have a lot of publicly available data and they sort of falls into two different camps. So there's a camp that says that we should be just scanning as many people as we can and then sort of by necessity not collecting too much data on any one subject, right? So that's what we call high-end, sparsely sampled. And then there's another camp that feel that we're better using our resources if we scan fewer people, but scan them many times under many different conditions to try to get a handle sort of on more within subject variability. So that's the first question. And then the second question is, which populations should we be targeting? Should we be just trying to get a handle on individual differences in healthy controls? Should we be extending out into patient populations? If so, which ones? So to break each of these down a little bit more, this first question of high-end sparsely sampled versus low-end densely sampled, as with everything, there's pros and cons to both. So if we're talking about sort of this low-end dense sampling approach, dense sampling really helps us characterize the within subject variability, and that's really important because if we just scan people one time each and we're looking for things that differ across individuals, we have no way of knowing if the features that we're seeing in your brain today are just sort of because you didn't get much sleep last night, and if they would even be there if we scanned you again tomorrow. So it's really important to consider both within subject variability and between subject variability when we're trying to get a handle on what is actually a stable individual difference, but we can't do that with only one time point per subject. So just on a basic level, by densely sampling people, we can get a handle on this within subject variability. And this becomes important, so we know that many psychiatric illnesses are not necessarily stable. People have good days and bad days. People have symptoms waxing and waning. And so ultimately, if our goal is to understand mental illness, then getting a handle on this intra-subject variability is going to be really, really helpful. And so the first study to try and do this was this work by a researcher named Russ Poldrack, and he started this MyConnectome project where he scanned himself I think upwards of 100 times over a period of 18 months. And when they began to analyze the data, I know this is a little small, I apologize, but basically this was sort of one of the early findings suggesting that if you take one of these group parcellations, which is up here in the top row, so this is sort of a parcellation that was computed on a large group of people, it represents sort of an average map of region boundaries in the human brain. But with enough data on one person, you can compute this parcellation for a single subject, and when they did this, they actually started to see some fairly substantial differences between where this particular individual's node boundaries were drawn versus the group. And another interesting thing you can get out of these long sort of many session within subject studies is you can start to control for other factors. So this was just sort of like a fun, interesting one that Russ did, but he was scanning himself on Tuesdays and Thursdays. On Tuesdays, he scanned himself in the morning with not having eaten anything and not having had caffeine. On Thursday, he allowed himself to eat breakfast and have coffee, and so you can start to see this is sort of a network structure representation, and there's some, not massive, but certainly noticeable differences in his functional connectome sort of modular structure, depending on whether he'd had caffeine or not. And, these are things that we don't necessarily control for or ask our subjects, and so it's just a demonstration that there can be this sort of within-subject variability. And then even if we don't necessarily have a lot of subjects, if we have more than one, so this project of Russ's actually inspired another project that was done down at WashU called the Midnight Scan Club. And this time they had 10 individuals that they were densely sampling, and they found a very similar thing. So up here are the group parcellation maps, and then each of these 10 brains is a different individual in this data set. I think they were sampled 10 times. It was a 10 by 10 design. And they observed that each individual now has a slightly different map that's different from the group and that's different from other individuals. So this, is just confirming that basically the more data you have on a subject, the more likely you are to be able to pull out stable individual differences on that person. So that's sort of a plug for this low-end densely sampled approach. I mean, it kind of goes without saying that in an ideal world, the best approach would be high-end and dense sampling, but typically you're limited by logistics and scan resources and things like that. So these would be some arguments for scanning not that many people, a large number of times each. But what are the arguments for taking the other sort of perhaps more standard approach, which is just to sample as many people as you can, but only sample them once? There's clear advantages here too. So the first one in my mind is just sampling a wider distribution of behaviors and phenotypes. So if we want to try and relate the in-scanner measures to the out-of-scanner measures like symptoms or performance on tasks and things like this, we just need a certain distribution. And this was just a quick illustration that I made to show. So this Midnight Scan Club, where they had 10 subjects, they had their subjects complete many of the same out-of-scanner measures that the Human Connectome Project did. But there's just no comparison in terms of this is only 10 data points. So even a measure like this, I'm not sure if you guys can see this, but this is a picture sequence. It's sort of a cognitive memory task. So they, each of their 10 subjects actually scored fairly differently, but, you just have nowhere near the amount of data that you would in a sample of 1,200 subjects like the HCP data has to try to get a handle on models and relating the brain features to real-world behavioral features. Same thing, this is a reading test. If you have a high-end data set where everyone has sort of the same measures or roughly the same measures, you can do this type of cross-validated model building. So you can divide your large sample into a training set and a test set, and then try and build models and see how well they generalize. It's harder to do this if you have less data. So this is just another advantage for high-end sampling. And it's potentially more realistic for real-world applications. So it would be hard to imagine a scenario where we needed to sample someone for sort of 18 months, really densely in order to compute some sort of clinical biomarker on them. Maybe, but in reality, we're probably going to have to get to a place where we can get sufficient information out of, let's say, an hour's worth of scanning. But there's potentially a way to combine these two approaches. So one interesting way to go might be to sort of build a model based on a small number of subjects that are very densely phenotyped, so that way you can try to separate the between-subject variability from the within-subject variability, train your model to recognize those features that are most stable within individuals, and then apply that model to a larger group of subjects that are more sparsely sampled, but now you sort of have the advantage of knowing which features are most important. So it's not that these are mutually exclusive, but certainly from a practical logistical standpoint, when we're deciding where to allocate our scan resources, these are some things to think about. Yeah.
Do you think either of these is, I guess, logistically easier or better monetarily almost? Because I feel like it would be more difficult to get people who are willing to come in 20 times to be scanned. So I don't know if you think that's a significant enough difference for that to be a positive for high-end sparsely sampled, or?
That's a great question. I'm going to repeat it for the recording. So Chandler's asking if there's sort of a trade-off in terms of if you do these low-end densely sampled studies, you're almost by definition going to be studying a certain population. So it's people that are willing to come in, 10 or 50 or 100 times for a scan. And in fact, this Midnight Scan Club data, the reason it's called the Midnight Scan Club is because they scan themselves. So it was 10 researchers at WashU that were willing to come in at night because that's when the scanner was available and cheap. And so they collected all this data on themselves. And you're right that sort of is restricting your population to a certain type of person that's probably pretty highly educated and willing to sort of come in and do this. And that is a big problem. And so I think that's definitely something to be aware of, and you're probably going to have just less phenotypic variability overall with that type of thing. There are these even larger initiatives like the Biobank that's trying to longitudinally follow, a very large sort of nationwide sample, but those are huge initiatives. So that is, it's another trade-off, and it's a good point. It's something to be aware of. Cool. Okay, so you've decided if you want to scan 100 people once or 10 people 10 times, who are you going to scan? This actually gets back to this same point. So patients or controls? Well, ideally both. But let's say you want to do patients. Well, which diagnosis or diagnoses should we be targeting? A lot of the patient work today is still done as sort of a dichotomy between control and then some kind of patient. Let's say it's depression. But there can be a lot of confounds there, so typically depressed people are on medication, or they've just been living with depression for a certain amount of time, and so I think some people are starting to take that into account and potentially scan two different diagnostic groups, so have a depressed group and a bipolar group, and try and see what is unique to each diagnosis versus what is just some sort of shared pathology that just comes with any diagnosis of a mental illness. But I and many other people before me are arguing that maybe it doesn't make so much sense to try and stratify people into these diagnostic categories beforehand, because we know that in a lot of cases these categories are hugely heterogeneous. They don't necessarily predict things about outcomes or response to treatment or things like this. Even just within depression, there's just so much heterogeneity in terms of which symptoms people experience. And so the intuition is that maybe we're not carving nature at its joints, to use an overused analogy, but maybe these are not necessarily biological categories, but they're ones that have been imposed based on symptoms and sort of historical clinical conventions. But there's this idea that if we sort of take a step back and go back to the drawing board and sort of just recruit, broadly, and then try and in a data-driven way sort of redraw these boundary lines between different conditions, that we might get a little bit more traction on finding what's actually biologically meaningful, or sort of which types of things are reflected in a biological way rather than just in a sort of surface level symptom-based way. And so this is a really nice illustration from Tom Insel back when he was here at NIMH. So this is the sort of RDoC idea, stands for Research Domain Criteria. It's been pretty influential, arguing that we should try and treat all of this pathology as more of a continuous dimensional thing and measure specific biological factors rather than just sort of take diagnoses at face value. But how do we sort of know, if we're not going to target any specific diagnosis, how do we recruit and sort of be assured that we're going to have at least some people, towards these more extreme ends of the spectrum? There's this idea that using a help-seeking model can sort of enrich your samples for pathology. So what I mean by that, this is actually a really nice example from the Child Mind Institute, who's in the midst of recruiting a 10,000 child cohort where they're doing lots of neuroimaging and behavioral characterization. And they use what I think is this really cool model where they just advertise basically all over New York in the subways, on the buses, everywhere, and they're sort of asking this very broad question, which is, are you worried about your child? And most parents are, right, in one way or another. But the idea is that they're not asking specifically about, attention symptoms or anxiety symptoms. They're sort of broadly trying to get people that, think that their child might have some sort of struggle with mental health related things. And they're offering for them to come in and get this workup and as part of this be part of the study. So this is sort of a way that you could target a broader population and not necessarily focus on any one disorder, but still try and get people in the door who, might be representing some of those more extreme ends of the phenotypic spectrum. This will be sort of a running theme throughout the talk, but in general, I think we all know, but it's hard to do in practice. So longitudinal studies are really going to be key. So this idea of recruiting patients while they're still controlled, so to speak, following people over time, seeing what what people at risk might look like, but this is, again, logistically challenging. So, assuming you're trying to do something cross-sectional, it might be a good idea to sort of target broadly, transdiagnostically, to try and pin down these biological categories. Okay, so that's sort of it for the subject section. So moving on to imaging, you've decided who you want to scan. They've shown up at your door. What do you do when you put them in the scanner? I'm going to try to go through this pretty quickly. So one of the questions actually that we first got a lot after we published the identification or the fingerprinting paper was, well, is this just a unique feature of the Human Connectome project? At the time, it was considered very high quality, state-of-the-art data. So do you really need data that's this high quality? And the answer is not really. It seems like at least this identification result is fairly robust, even at more standard spatial and temporal resolutions for your imaging. I'm not going to go through these in too much detail, but basically this was looking at these 4 graphs are different spatial and temporal resolutions. Here the y-axis is actually lower is better, so lower is higher identification rate, and really the main thing seems to be acquisition time, which is on the x-axis here. So the longer you scan someone, the better you're able to characterize them and the better your identification rates will be. This was an examination specifically of TR, or temporal resolution, where they were basically just mimicking a sort of temporal downsampling. So they're just taking basically every other or every third or every fourth, etc. frame and testing what that does to the fingerprinting accuracy. And they saw that it takes a pretty long TR, so to speak, for the accuracy to drop. So even at up to like 10, 12 seconds, just one frame, as long as you sample for a long enough time, you can get a pretty stable in the sense that it's at least different enough from other subjects to still characterize people. The thing that does seem to matter a fair amount is amount of data, which I've already kind of alluded to. So scan duration really matters. As you might guess, longer acquisitions are better. So longer acquisitions lead to higher reliability within subjects, as well as higher identifiability across subjects. So this is the sort of within versus between subject variability distinction here. A higher sampling rate, this one's kind of interesting. So if you just take more samples, that can actually make up for a shorter scan duration. So it's kind of easiest to see in the bottom graph here, but here they're sort of artificially temporally downsampling with 2 different approaches. The red line here is if they're just randomly removing frames. And so you can see that that doesn't have too much of an effect. So even if you cut out 50% of your data, as long as you're just randomly choosing frames to drop, you can still get pretty good within subject reliability. Whereas if you're removing your data by truncating, so just lopping off, the last X minutes of your scan, that starts to degrade really, really quickly. So there is something about just having the person in the scanner for a certain amount of time. We might hypothesize that this is related to something like dynamics or brain states that they're traversing. We just need to capture enough of those to have a stable representation of people. So to the extent that people can tolerate it, it's nice to have them in the scanner for as long as you can. Okay, what should we have them doing in the scanner? This is a really big one. So I've been talking a lot about resting state connectivity. This was sort of, a lot of these functional connectivity approaches were first developed on resting state, but it turns out that you can also do them during task, and you might want to do them during task because there's some evidence that tasks actually increase the signal to noise when we're talking about these individual differences. So this is a results matrix here where we were replicating those identification experiments. So basically, and this is back to Human Connectome project data, this is essentially just across the rows and columns, it's the different pairings of scans that we're trying to match people across. And the point here is that there's a lot of variability in how well we're able to identify people depending on which of these pairings that we're using. And one thing that I'd like to draw your attention to, so now we've expanded to a larger subset, so our rates have dropped a little bit. We're still doing pretty well between the 2 resting state scans, which were again on different days, so we're able to identify people between rest 1 and rest 2 with around 70% accuracy, whereas again, chance here is less than 0.01% or 0.1%. But the 70% is actually not as high as some of the task-to-task pairings And this is interesting because none of these tasks are the same. So we're actually doing better when we match people, for example, between the working memory and the gambling scan. We're getting up to 92% accuracy than between rest, which in theory is the same condition both times. But resting state is so unconstrained and potentially subject to these other factors, these within-subject fluctuations that we don't have a good handle on, that we're actually seeing that the presence of some kind of task, really any kind of task, is serving to sort of stabilize this individual functional architecture in a certain way. So is resting state really the best scan condition to use? Probably not. Another interesting thing that I've and others in the lab have gotten interested in lately is using these naturalistic tasks. So these are things where you have subjects, let's say, watch a movie in the scanner or listen to a story. It can be something really abstract, like this video here. This is called Inscapes. My citation is cut off, but it was developed by Tammy Vanderall at Yale, actually for pediatric imaging, just to give the kids something to look at in the scanner. And she observed that there were huge practical advantages to this, so kids actually moved a lot less and they stayed awake better when they had this sort of abstract video to look at. This was another, this is actually a clip from Ocean's 11. So, these are two very different movies in terms of their content, but this is another way, this is another type of scan condition that you might think about. And the interesting thing with these naturalistic tasks is that when people first started using them in imaging experiments, people noticed that they really tended to synchronize a lot of brain activity across individuals. And so when we first became interested in these, we were wondering what that would do to our individual signals, so to speak. So if we sort of give everyone the same dynamic, engaging stimulus to watch, are we going to wipe away, all of the individual variability that we care about because everyone's going to look so similar to one another? But when we looked at this empirically, at least in our sort of functional connectivity-based identification framework, we actually observed that the identification rate in these naturalistic tasks was just as good, if not better, than rest. So here's our connectivity identification accuracy matrix here. You can see these squares along the diagonal are between two scans of the same condition. So this was a test-retest study across 2 different days. And we're doing really well. We're doing well, much above chance, both within video, so rest 1 to rest 2, Inscapes 1 to Inscapes 2. But we're also doing really well in these off-diagonal elements too, suggesting that we're able to match people between resting state and watching a movie pretty well. Even though everyone looks more similar in the movie condition, they still look enough like themselves that we can pick up on this individual signal. Another interesting and important feature, I think, of considering using task-based as opposed to resting state data is that also, just based on empirical observations, that if you feed a behavioral prediction model data from a task study, it actually does better in a lot of cases than if you feed it rest. So I had told you before that we were able to predict people's levels of fluid intelligence just based on their resting state connectivity. But it turns out that when we went back and tried feeding the model connectivity from, for example, working memory or a language scan or even a motor task, really anything, we were able to generate more accurate predictions of behavior. And so that's what this graph here is showing. Here on the rows, we're varying the input to the model, so functional connectivity calculated from working memory, language, gambling, etc. These are 3 different out-of-scanner behaviors that we're trying to predict. And the main takeaway here is you can see that the 2 resting state scans are doing the worst. So they're still doing above chance, but they're not doing nearly as well as if you just give the brain something, anything to do while we're trying to generate these predictions of behavior. And the analogy that we like to use here is sort of like a stress test. So for example, if you think about cardiology, maybe people's, you can't pick up on abnormal heart rhythms when people are just sitting in a chair, but if you make them walk on a treadmill and actually elevate their heart rate, then you might see these abnormalities come out. And this is sort of a similar thing that we're thinking about with the brain data, is that if you actually give people something to try and do, you can pick up on this individual variability a lot better. That's true. Yeah, so the question, or the observation, is that the working memory task here, which is in the top row, doesn't actually do the, well, yeah, it does. It does better than the other task. Yeah, so this is a harder one to predict, and these are all, so this was sort of a different test of working memory that was done outside the scanner. So the working memory task in the scanner, I think, was an NBAC. Outside the scanner, it was like a card sort kind of a task. And so I think this one is maybe just less predictable across the board. But we also tried predicting, so there's at least some of these scans have behavior associated with the scan itself. And it's not actually always the case that we can best predict the in-scanner behavior from the concurrent task, which is really interesting. And I don't have a great answer or summary to that, but it's, yeah. Yeah.
So I'm with you all the way on task-based analyses, but did you ever look similarly if you had a REST 1, REST 2, and see how similar they were with an individual? Did you look working memory 1, working memory 2?
That's a great question. We would have loved to do that. Unfortunately, the HCP didn't collect that, so the only thing that was repeated was the REST. So in this particular data set, which was nice because it was so large, we weren't able to look at that. There are other data sets that have sort of that test-retest on different tasks, so that the naturalistic viewing one I just presented, for example, was one of those. But yeah, there are other data sets out there that have the same sort of traditional cognitive task run twice, I believe, but it's smaller numbers of subjects. So I'd have to, I don't know off the top of my head. Yeah, actually there was a recent paper on reliability of activation during task, which seemed to be not as good as you would hope. But I guess and that's certainly, I think not trying to discount those findings. That's a huge area of important research. I think that there is sort of a subtle distinction here between just something that's super reliable within a person. So if we scan you today and get 7, do we get 7 again tomorrow? Or do we get 6? Whereas in someone else, we get 100 and then we get 101. So I guess the distinction I'm trying to draw is something can be a little bit less reliable within subjects, but as long as each person doesn't change so much so as to look like someone else, there's still room, I think, to pick up on something that's stable about an individual. And so it really just depends on your application. It depends on your ability to sort of know how much variability is too much, if that makes sense. Yeah.
Different question. So regarding this with stress test hypothesis, I think there are ways that you can turn fMRI into kind of you can put it into wattage, right? Like how hard is the brain working per unit time? So HLBI and NINDS have done a lot of this called brain energetics, right? So I think your theory would predict that the harder the brain is working, the better it should predict things. You consider that?
Yeah, that's an interesting way to frame it. I've thought about it like that a little bit. I think that the way that I've thought about it more actually is because that's a lovely framework, but then you sort of get into what is really how do we get an absolute activation? Does that differ and things like that.
Taliban has a paper that does that. I mean, it's crude, obviously, right? I mean, you're making an estimate, but you can sort of take an fMRI signal and make an estimate.
Yeah, no, that's cool. I mean, I should certainly look at, or the delta even between rest and a task,
exactly.
getting at it that way. I mean, the way that I've actually thought about it more is this idea of conditions that make people look more similar versus less similar, and so there's this sort of paradoxical result that I skipped over, which was that task conditions that sort of bring everyone together overall in this connectivity space, you'd expect maybe that those would obscure the individual differences, but actually we see a positive correlation between how similar people look to one another, but then also how identifiable they are. So,
noise, not
right. So you're sort of, so the way I think about it is you're constraining you're basically projecting everyone onto this shared space, and so even though everyone's coming closer together, then the actual differences between them become more meaningful. Yeah, you're boosting signal that's actually neurally related and cognitively related, let's say. Yeah. The final question in my sort of imaging block of questions is this question of multi-site studies. So getting back to the question of N and high N or low N and how many times do you sample. Obviously, recruiting people from multiple sites and getting sites together and coordinating can help increase your N a lot, but it can also introduce some added variance. So this was a result from a couple years ago. showing that this was 8 subjects that traveled to 8 different sites, and we were looking at how much connectivity could be explained by these different factors. So you can see happily a lot of the variance was accounted for just by subject, which is what we like to see. But there were certainly some site and scanner manufacturers, so this is Siemens versus GE. There was some variance accounted for by these 2 things that were sort of regionally specific, perhaps unsurprisingly, OFC, a classic sort of dropout region. But ultimately, if we want to create a biomarker that's useful in the real world, it should hopefully be robust to site. So it shouldn't matter, where we're scanning people. And just as a quick shout out to work from Dylan Nielsen and Adam Thomas, it does seem that there's some promising techniques to try and harmonize data across sites. So Dylan applied this algorithm called COMBAT, which is a sort of a Bayesian framework for trying to remove some of these side effects. And this is really, really small, but the only point I want you to see here is that all of these rows here are different features of connectivity, basically different brain metrics. This was before he applied the algorithm. The blue stuff is how much site accounted for the variance. And after he applied the algorithm, all of that blue stuff was pretty much taken away. So that's a good thing, and it suggests that we can actually combine data from multiple sites to boost our power and boost our statistical rigor. But we might want to use these types of techniques first to account for that added variance. You've decided who you want to scan, you've scanned them. What else do you want to know about them? What are the sort of real world metrics that you want to try and link their brain imaging data to? And so there's a couple questions here, sort of broad categories of behavior. We have self-report, or what I'm calling behavior. By behavior here, I mean sort of task-based performance, so not things that you're asking people to sort of consciously report, but actually have them perform a task and extract things like performance or reaction time or these sort of more implicit variables. And, historically, there's been some recognized issues with self-report. So self-report are things like questionnaires, personality surveys, things that you're asking people to sort of explicitly assess themselves. We know that these are subject to demand characteristics, meaning, if I come participate in your study and I think I know what you want me to say, I might say it, or, people being overly positive or overly negative when they assess themselves. So there's all of these sort of biases that come along with self-report. Traditionally we think of behavior as being less biased. It's harder to sort of game your performance on a task. You can still do it, but it's less clear how that would be as biased. An interesting recent study actually found that self-report might be more stable within individuals and more variable across individuals, which is what we want, in theory, for looking at trait-like individual differences. So this was a study of cognitive control, all these different measures, all these different ways we try and measure cognitive control, both with tasks and with surveys. And so everything in orange here in this first column was a task-based behavior, and everything in blue here was a survey. And basically, these are reliability coefficients. Each dot is a different study. And so the closer to 1, the more reliable the measure was. And so actually, these authors observed that the self-report surveys were actually more reliable overall than the task-based sort of behavioral measures. And they even potentially showed these characteristics that we look for in individual differences. So we want to see more between subject variability, that's the blue bar here with the survey, so that's higher between subject variance and actually lower within subject variance. So, the takeaway here is it's complicated. I think ideally assessing traits and characteristics with a battery of instruments rather than a single one is going to be really important. And then, of course, it's important to keep in mind that our gold standard at the end of the day is going to be predicting real-world outcomes, right? So if I just give you a pencil and paper test on the same day as I give you your scan, and then I try to use your scan to predict your pencil and paper test, ultimately I could just look at your pencil and paper test, right? The brain data is just going to be a noisier version of the behavior, but where the brain data can really help us is predicting things that we can't know on that same day. So like whether you're going to go on to develop some kind of illness or whether you're going to respond to some kind of treatment. So again, getting back to the need for longitudinal sort of real world assessments that will tell us just how much value added there will be by these types of functional imaging biomarkers. Getting on to our second column of stuff, I swear these next 2 columns are shorter, so bear with me, there's a lot on the acquisition side. On the analysis side, there are also still many, many open questions. So the sort of biggest one in my mind is one that's received a lot of attention over the last couple of years, which is basically features of the data and level of analysis. So where do we want to be when we're trying to pull out this individual signal? How do we want to analyze our data? So functional connectivity has gotten hugely popular, but we can still do, sort of classic task-based activation studies. And maybe we were a bit too quick to jump over to functional connectivity. Maybe there's more information to be had in the activation. And then specific to the connectivity stuff, there's debates going on right now about whether it's sort of the connections between the nodes or the parcels or the parcel boundaries themselves. So to dive into this a little bit more, I think this current focus on functional connectivity really stems from the explosion of resting state and large-scale data sets. Functional connectivity is a highly dimensional way to analyze your data, so you end up with many, many features to try and relate to behavior, and that can be a good thing as long as your model is appropriately accounting for that dimensionality. But I think, maybe some of the more pure psychologists would agree with me here. I think, people have been doing individual differences with fMRI for a very long time. And in fact, this is a study from 2003 showing that this fluid intelligence measure, basically the same one that we are predicting in the HCP dataset, is also, this isn't a prediction, it's a correlation, but they found this pretty strong relationship between activity during a working memory task in this lateral PFC area and people's score on this fluid intelligence measure outside the scanner. So there's still a lot of individual information embedded in just the task-based activation signal, and this has been known for a long time. And so maybe some sort of combination of functional connectivity and activation is where we want to be. This is just another more recent example on the HCP data predicting intelligence from the sort of difference between frontoparietal activity and default mode activity. So these are regions that are defined based on connectivity, but they're actually looking at their activation and showing that there's meaningful individual signal there too. So again, maybe some combination is where we want to be. And then there's other sort of more non-traditional approaches, such as inter-subject correlation. So this is most typically applied to a naturalistic paradigm, like I was talking about with the movies and things like that. And so looking at sort of this sort of in-between, or maybe this third qualitatively different thing from connectivity and activation would be synchrony across people, so how well your brain synchronizes with someone else or fails to do so, and looking for individual signal in that. So again, I'm leaving you with more questions than answers, but these are all things to think about. And I think we're all quick to jump to functional connectivity when it involves resting state, which makes sense, but considering these other techniques that have been around for a long time might also bring us more power. Okay, so there's this, also I mentioned there's this big debate right now about whether it's the functional connections, so those correlations between average time courses and regions, or whether it's actually where we're drawing the node boundaries themselves. And so people have observed that imposing a group atlas, like the ones I've been mostly talking about, can obscure individual differences in node boundaries. So we saw with both the MyConnectome dataset and the Midnight Scan Club dataset that when you have enough data on someone, you can actually calculate an individual parcellation, and its little boundaries are going to look different from the group average. And people have now taken this a step further and said that the node boundaries themselves, so the exact placement of where and how large people's functional regions are, can actually contain meaningful information. So this was a study that came out in eLife last year. This is hard to see, but if you look here, this gray outline is the node boundary from the group parcellation. And these two brains here, so this was the average across the half of the people that scored lower on many of the cognitive and other sort of lifestyle metrics. And on the right here is the half of the subjects that scored higher. And they have this pretty subtle, but apparently replicable distinction in that the group with the higher scores had, when you run a parcellation on them, they actually have this little bit here that's extending further posterior. And so the argument here is that, well, maybe this group atlas was a better fit for one group or the other, and so when you start averaging all of the voxels within this node, if it's a better fit for some of your subjects than others, then what you're looking at is not so much long-range connectivity between, for example, this gray node and then this other gray node up here, but you're actually just looking at how well this gray node fits the data in the first place. And the fact that there is sort of a behavioral difference between groups of subjects in how they parcellate is certainly interesting and provocative. And this has been sort of conceptually replicated and extended by some work from Thomas Yo's group, where they took Human Connectome Project data, and they actually came up with a Bayesian framework for running an individual parcellation on HCP subjects. So only 2 scans worth of data, but they were able to get a good individual parcellation fit, and then they were actually able to predict a number of I'm so sorry that my references are cut off here. This is a really big bummer. If anyone wants the references to these papers, please talk to me afterwards. But essentially, they were able to predict many of these HCP behavioral measures outside the scanner by just looking at the boundaries of an individual's parcellation and trying to match that to a subject in the training set that had similar boundaries. So it does seem that these node boundaries do contain meaningful individual information. I'm going to skip this. Whenever we talk about connectivity, we have to mention head motion. I keep thinking people know this, and then I get more papers to review where they haven't considered it. So just a quick plug that many behaviors and phenotypes are correlated with head motion, and this can be a really big confound. So this paper, just again looking at Human Connectome Project data, showing that just how much people move their heads in the scanner. People that have higher cognitive performance tend to move less. People that are sort of less healthy in some way or have lower cognitive performance tend to move more. There's also a patient effect here. So in general, this isn't always true, but in general, a lot of times patients will move more than controls, and both children and elderly people will move more than sort of young adults or younger adults. So this is just something to be aware of. There's ways to mitigate this. So the first thing to do is just check for it. So you can't control for it if you don't check for it. You can consider excluding very high motion subjects. There's a lot, you can't do everything, but there's a lot you can do with appropriate preprocessing. So there's been a lot of work looking at sort of the best preprocessing pipelines to try and mitigate the effects of head motion. This was a paper from Ted Satterthwaite's group showing every square here is a different preprocessing strategy, and basically as you add more and more stuff, the number of edges in blue are the number of edges or connections that correlate with head motion. And so, by the time you get here, you're certainly not perfect, but you've removed a lot of the influence of head motion on connectivity. You can also use motion as an explicit covariate, obviously, in your prediction models and things like that. Okay, we are almost done. So finally, there's some things to think about when we actually think about applying this stuff in the real world. And another question that we get a lot that I think is super interesting and people have started to make some progress on is how mutable or how changeable are these individual signatures. So we've sort of shown over a period of 1 or 2 days, in Human Connectome, where people are coming back on contiguous days in different brain states, that we can do this fingerprinting, so to speak, and we can predict these behaviors that are, again, concurrent with the scan usually. So it's a test that they did, on the same day or at a very similar time as they did the scan. But there's all these open questions about how this changes. So, we know that you're probably not born with your functional connectome fingerprint and then it just stays stable your whole life. We know that this must probably develop somehow. We have an intuition that it would change, if you became sick or even if you learned something. So there's all of these things that can change the brain, and we know that that's true. And so figuring out how much of this is mutable and how we can sort of pin down these within-subject changes versus these more stable trait-like factors is an area of a lot of interest right now. And so there's been a little bit of work on this so far. So there's one study came out suggesting that identification is possible across sessions separated by a period of years. So this was, they were using 4 different data sets, and they all, interestingly, had sort of different aged populations. So from teens up to older adults, and they were scanned at least twice, sometimes 3 times in the case of many of these data sets. The point here is that ID rate is on the y-axis, and then, for example, identification between sessions 1 and 2, sessions 1 and 3, sessions 2 and 3. The gray bars are chance, so all of these are well above chance, and these are scans separated by, on average, about 1 to 3 years. So there's some stability, at least on that time scale. Interestingly, there wasn't a strong correlation between how many years between the scan and how much the subject looked like his or her previous scan. So some of these showed sort of a decrease, which is maybe what you would expect, but in large part, these scatter plots are all pretty stable. So again, what would this look like 10 years, 20 years out? We don't know. But it's interesting that the time between scans, at least on this sort of 2 to 3 year time scale, didn't seem to affect things too much. Another place people have looked at this is in even younger developmental samples. So there was this interesting paper that came out here showing that this is age along the x-axis. It goes from 8 to about 25 or so. This is a metric of connectome distinctiveness that they came up with. So, the more distinctive, the higher the individual signal. And they find this sort of pretty striking increase in distinctiveness with age. Interestingly, it seemed like females sort of reached this distinctiveness earlier. So females are in the orange curve here and males are in the blue curve. In this same paper, they also saw that the process of achieving a distinctive connectome, so to speak, was delayed in the kids that had some sort of mental illness. So in black here are the healthy individuals, and in red are people with increased clinical symptoms. I think in this data set it was mostly sort of ADD type symptoms, but you can see that the red line kind of lags the black line, suggesting that these individuals had a slower maturation of their connectome distinctiveness. But all of this data, this was a really interesting study, but it was all cross-sectional, so they only had one time point per person. So these are not actually growth curves within individuals, they're just sort of comparing people that were different ages at the time of the scan, and so what this suggests is that we really do need longitudinal data. And a quick plug for this new data set that's just starting to emerge called the ABCD data, or the Adolescent Brain and Cognitive Development Initiative. This is a huge scale initiative. They're recruiting, I think, 10,000 kids starting at age 9 or 10. They're following them for a period of 10 years. They get workups every year. I think the imaging is only every other year, but still it's going to be a huge treasure trove of information to try and really pin down these within individual changes in a longitudinal framework. Okay, that's about all I have. I think, looking ahead, if it's not clear, I think we have a lot to do before any of this stuff is going to be ready to be rolled out, in the clinic or in the classroom or what have you. But I think there is reason for optimism. And one question that I get a lot is sort of, actually not so much from researchers, but from people sort of outside in other professions, is sort of the ethics of this. I don't have a great answer to this, but anytime that we're predicting something about an individual, we risk things becoming self-fulfilling prophecies, so to speak. So I think that there is a strong overlap here with sort of bioethics, and in a theoretical future, if we had a machine that could perfectly predict something about a person, would we want to use it? Who would have access to that information? There's all sorts of things to consider here, and I'm not an ethicist or a philosopher, but I strongly believe that we should be thinking along those lines, and we should be preparing for some sort of a future where that's true, and making sure that anything we develop is used for good and not for harm. So if you're interested in some further reading, these papers on the left are ones that, the last one is mine, so it's a plug, but the first three are ones that inspired a lot of my thinking on this. They're really good overviews of sort of where we are in the individual differences world, and I've sort of plugged many open data sets along the way, but there's a ton of amazing publicly available data out there that you can play around with on your own or in conjunction with your own homegrown lab data sets to sort of test these ideas of generalizability and things like that. And so I'd just like to acknowledge my current lab as well as some collaborators back at Yale. And if you have any questions or you want to chat about this stuff, feel free to email me. Same goes for people watching online, and thanks for your attention. Yeah, that's a really good question. So the question was, so I had shown a slide of people demonstrating that sulcal depth and cortical thickness are also most variable in these regions where we see the most variability in the functional measures, and have people tried to relate features of anatomy to diagnosis or other things like that. There's a fairly sizable literature on like voxel-based morphometry, for example, and how that relates to things like fluid intelligence. It's funny, I think that's kind of, I don't know if anyone's actually run that on HTTP. That was sort of popular a while ago, and people were working with smaller sample sizes, so it was harder to tell, how well those models would generalize. Certainly, there's been papers reporting correlations between things like cortical thickness and BBM type measures, and these cognitive measures. There's also studies, schizophrenia, cortical thinning in PFC and various subregions of that. I think it's a weakness of our field that there's not a lot of people sort of combining the anatomical and the functional approaches. I think at the end of the day, probably a lot of this stuff is reflected in anatomy, but I think the turn to function was sort of inspired by the lack of super robust anatomical measures. So that's kind of an unsatisfying answer, but as per usual, we'd probably get more power if we tried to combine these things.
I'm surprised you think the anatomical measures aren't robust. I mean, I think they have lots of limitations in that they're kind of, they're not four-dimensional, they don't measure strategy or change task. But I think they are robust by being very replicable.
Yeah, no, absolutely. To clarify, I think that when a lot of those studies were first being done, we didn't have the sample sizes to determine if the association between the anatomical measure and the behavioral measure was robust. So I think the test-retest on anatomy is much higher. I mean, that's an empirical fact, is that the test-retest on anatomy is much higher when you're just looking brain to brain. But there's a question mark about the I mean, as with any of this stuff, but the point I was trying to make was, I think, a smaller one, which is just that a lot of those classic studies were run on, n equals 20 and relating cortical thickness or whatever to behavioral measures, and so to try and see whether that holds up. I know people are working on it. I'm not as aware of the structural literature as I am of the functional stuff.
I think a lot of them are not holding up. So I just saw a few weeks ago that the relationship between hippocampal volume and depression doesn't hold up if you get a really large sample together. And that was pretty well established. So I think a lot of those are going to start cracking.
Fair enough.
Thank you for your talk. It's an amazing field. And do you think it's important to take into account sets as a variable that will be implicated in these individual differences? Do you think it's important to take into account sets?
Yeah, absolutely. In fact, one of my slides I got cut. So there's been some work showing that if you stratify, especially I think developmentally, which is interesting. So there was a study showing sort of cross data set prediction between Human Connectome project data and this Penn developmental sample, where they were predicting sort of working memory in both, but they were able to train a model on one data set and apply it to data from the other data set and actually get some prediction accuracy, which is amazing in terms of generalizability. But the point was that they were also showing that if you train models separately on males and females, that you could get better, there were slightly different features that better predicted working memory in this case in boys and girls. So yeah, I mean, I think all of this stuff plays a huge role. I think that's the value out of these large data sets is now, you can afford to sort of stratify your sample that way and not lose too much power. But yeah, there's definitely those effects, and they should be taken into account.
So the research you showed that like some of the temporal regions and things that were kind of more recent phylogenetically, I'm surprised, is the hippocampus not coming up as an area that's very different? It's implicated in so many psychiatric disorders and things like that, but that would be very old, obviously.
Yeah, that's a great question. I mean, a lot of the maps that I showed you are cortical only because for some reason a lot of people only analyze the cortical sheet. That's actually a pet peeve of mine. I try to do, there's a lot of pros to surface-based analysis, but that really only makes sense for the cortex, and somehow a lot of people have gotten away from even looking at subcortical, including amygdala, hippocampus, as well as cerebellar data. I personally have observed a ton of individual difference behavior relationships in the cerebellum, and I think, I don't know the hippocampal literature in particular as well, but I do think it's a shame that people ignore subcortical structures and cerebellar structures. It's amazingly pervasive, so you see a ton of figures that are cortical only, and I think especially when you're talking about connectivity, I mean, so much of the large-scale dynamics of cortex are driven by thalamus, for example, and loops with basal ganglia and hippocampus. So I don't have a great answer, but I would agree it's probably pretty important. We should try to look at all of our data. Cool. All right, thanks so much, guys.